Good morning and welcome once again to StudentSOS, or indeed a very warm welcome to you if this is the first video that you've watched in the series. Please do take a look at the videos we've already released. We are a new YouTube channel that started only 3 or 4 weeks ago, but we seem to be growing in interest, and so please hit that like button if you like what you see today. Take a little tour of the videos that we've already posted. We work with 11 plus through to UCAT and BMAT students, and my intention is to put video content up that is useful to all of those groups over the coming months and years. So please hit that subscribe button if any aspect of from 11 plus education all the way through to medicine applications, and particularly A-level biology at the moment, hit that subscribe button to join us. Please also consider taking a look at our Patreon. It is a £3 a month currently subscription, and it offers you discounts on one-to-one tutoring. It also offers opportunities for you to join live events, which we'll be doing several of over the summer, and all kinds of other sorts of content. If you have any sorts of comments you'd like to leave below that relate to content you'd like to see or you'd like more of, or indeed suggestions for what you'd like us to do in some of those exclusive content Patreon sessions, please leave a comment below. We read all of your comments and we'll reply to them, but we're really keen to offer you the service you want. The Patreon is really great because it allows us to keep offering free content on YouTube. This is a little picture which you'll be bored of me talking about now, but I like to have this as the start of the video. Now, this was a card sent to me by a very good friend, so I thought it was very apt for the YouTube channel. Today we're going to take a look at the second part of carbohydrates as biological molecules, and specifically therefore at polypeptides, their structure and how it relates to their function. So, structure and function links. There are two main functions for polysaccharides. They're for storage of energy, or they act in a structural manner. Now, I mentioned before, and here it says at the top, I make no apology for the fact that I consider this a dull topic. It's not my personal opinion, just that students find it very dull, and sometimes as a result quite tricky, and they don't spend enough time on it, and consequently the exam boards, all of them, tend to really love asking questions about polysaccharides because they assume, quite rightly, that most of you haven't learnt them properly. This is actually not that difficult. It is not the most exciting content I teach. Let's start with storage of energy. So in animals, we store energy as glycogen, mostly in the liver, but also in the skeletal muscle, ready for immediate use. And we store as starch in plants for the same reasons. So starch granules are stored in plants, so there is an immediate source of energy should there be none available as glucose. Glycogen does the same in animals. Their structure, well, they're compact. It's just glucose in long chains. There is some branching, so a myelopectin in glycogen, and there's some unbranched material, and that's amylose. So the idea of this is that we can do a single enzyme catalyzed reaction to remove a single glucose molecule at a time. If I have a branch structure, I can do many of these in a single enzyme catalyzed reaction all at the same time and release many dozens of glucose molecules at the same time. In a future video, we'll take a look at homeostasis of glucose, and we will have a look at exactly how this is controlled by the liver and the hormones glucagon and insulin. Both GCSE and A-level students should have heard of both of those. So, structural polysaccharides, primarily we'll take a look at cellulose. I would like to point out at this juncture that a polysaccharide type molecule called peptidoglycan has amino acids in it as well, so it's not a polysaccharide in its own right, but it is used for bacterial cell walls. So peptidoglycan is also a structural component that contains polysaccharide but also polypeptide, and you will have also heard of chitin. You will almost certainly pronounce it chitin if you've read it and not heard it said, but this is what forms the structural exoskeleton of insects, and that is also polysaccharide based, but you don't need to know more details about those two. They are referenced in your textbooks I think in all A-level texts. That's certainly true for OCR and AQA. So cellulose is used as a structural polysaccharide for plants. It's straight chains of this time beta-glucose. I did not mention that these guys are made of alpha glucose exclusively. So glycogen and starch are alpha glucose polysaccharides. In contrast to cellulose, which is a beta-glucose polysaccharide, it is exclusively beta-glucose, and it's connected with 1,4 glycosidic bonds. 1,4 simply means we use the first carbon in one of the glucose and join it to the fourth carbon of the one next to it. That's all we mean by a 1,4 glycosidic bond. So every other beta-glucose is flipped, 180-degree rotation, to prevent spiralling. So we need to keep that straight chain. And naturally, if you bind them next to one another, what they will do is coil as you increase the length of the chain. To avoid this, because we need straight chains for cellulose cell walls in plants, we use beta-glucose, which is then flipped every other beta-glucose by a 180-degree rotation, and that keeps straight chains. There are hydrogen bonds both within the chains and also between the chains, and they give strength to the cellulose, and it also helps prevent with the spiraling. The glycosidic bonds here are hard to break, unlike the glycosidic bonds over here, which are broken with a single enzyme catalyzed reaction. These glycosidic bonds in the beta-glucose are hard to break, and most animals, including humans, do not produce the enzyme cellulase to break them down. This is actually why we need plant material in our diet, because it acts as fibre, and we don't digest it. It aids something called digestive transit. So, things you need to know. The difference between alpha and beta glucose. I mentioned this on the previous video. Really important that you can draw alpha and beta glucose, and if they're drawn for you on an exam, you can recognise the difference between the two. Also, good idea to be able to describe in one sentence the actual difference between the two structures. Please comment below if you can do that. I'd be interested to know how many people can do that. Don't go checking it in the textbook first. Tell me that you know it as you watch the video. How structure is related to function in glycogen, starch and cellulose. So we've just taken a look at that. So cellulose structure is straight chain with hydrogen bonding within and between the chains, and that allows the structure to be straight and strong. This is really important because cellulose cell walls in plants are really the only way they maintain their stability. Unlike in animals who have all kinds of structures involved in terms of cartilage, tendons, ligaments, bones, extracellular tissue fluid, all sorts of things, plants only have cellulose cell walls. Whereas glycogen and starch, we wish to package as small as we can, so they take up the minimum amount of space. We also need to package glucose as either glycogen or starch, depending on whether you're a plant or an animal, because glucose will affect the water potential. We haven't looked at a video on water potential yet, another topic students don't enjoy. We shall do one of those very soon, but glucose will affect the water potential of a cell, causing osmosis. Now, glycogen and starch do not affect the water potential, and this is very important. But the compact nature of glycogen and starch means that what happens is we're able to release glucose as we need it in the cells and as the cell requires that, but otherwise it gets stored until it's needed. So we can quickly release the glucose for respiration and keep it safely and efficiently stored in the meantime. But that ends this second section on carbohydrates as biological molecules. We will move on to proteins next, but in the meantime, have a fantastic day. Please don't forget to like and subscribe to the channel, and indeed leave a comment below if you can answer questions I've asked in the video, or indeed if there's anything that you would like us to particularly cover with urgency. Thanks very much for watching today. Have a fantastic day. Take good care. Bye-bye.